Okay. Okay. So first of all, I'd like to welcome you all here today. And we have a really outstanding speaker, Patrick Seale. So Patrick completed his undergrad at McMaster University and his PhD as well, and studied developmental biology and myogenesis. And one of the interesting things that Patrick published during that time, he discovered PAC7, which was a driver of myogenic satellite cells. And so this was a pretty big discovery in the muscle field. And I think that really set him up to his postdoc with Bruce Spiegelman, where he discovered one of the first drivers of the brown fat differentiation program. And so this was PRDM16, which was published 2007. And that was just around the time when I joined Peter's lab as a postdoc. And we ended up following up on some of Patrick's work early on when I was a postdoc. And then Patrick went on to get a faculty job at UPenn, did really tremendous work on PRDM16 and following up in its role in driving beige fat thermogenesis and how that impacted metabolism. And has continued to really drive this field in understanding the role of progenitor cells in the context of obesity and type 2 diabetes. So Patrick is a professor in the Department of Cell and Developmental Biology, has received several awards, including the NIH Director's New Innovator Award. He's also a Searle Scholar and was recently selected to be the Weitzman Outstanding Early Career Investigator, and also the Linda Montage Investigator Award for a mid-career investigator at UPenn. He served on several study sections, some of the ones that we all probably have sent our grants to, including iPod and POMD. And we've probably served on study sections with him as well. So he's a quite thoughtful investigator. And one of the things that I always take away from Patrick is he's very clear and concise speaker and is always doing outstanding innovative science. So with that, let's give a round of applause to Patrick and welcome him.
All right, thank you so much, Claudio, for that overly generous introduction. It's great to be here and visit with you for the day. What I want to do today is really focus on two newer projects in the lab, and they're kind of distinct. So the talk is kind of going to be separated out into two different parts. The first one dealing with some of our recent work in trying to understand adipose tissue remodeling. And the second part, quite different, focusing on smooth muscle cells and atherosclerosis. And I'll try to explain how we got onto that topic. So just as an introduction, so our lab has had a longstanding interest in adipose tissue, and there's different types of adipose tissue. In the field, we think about white adipose tissue as well as brown and beige adipose tissue. All of these cell types we think play really critical roles in regulating systemic metabolism. They also have other roles, but in terms of regulating systemic metabolism, they have important roles. So brown and beige adipose tissue, these are thermogenic fat cell or fat tissue types, and they help in terms of promoting systemic metabolic health by increasing energy expenditure, and in doing so, they catabolize various substrates, including glucose, fatty acids, and other potentially toxic metabolites like branched-chain amino acids. And by consuming these metabolites, they protect the rest of the body, the rest of the organs from ectopic lipid deposition. It's important to point out, and important for this talk, to just note that white adipose tissue is also really important for preserving and maintaining metabolic health. And white adipose tissue, of course, is highly specialized for storing energy. And so having a healthy depot of white fat essentially it provides a safe storage place for energy and especially fatty acids, so they're not ectopically deposited in other tissues and thereby leading to insulin resistance and metabolic complications. And so what maintaining a healthy white adipose depot is critically important. And if you think about the processes that are important in maintaining adipose tissue function, adipocyte differentiation is important in this process. So the differentiation of new adipocytes from progenitor cells or precursor cells in the tissue is really important for healthy maintenance and expansion. In this state, there's also what we think about as being pro-regenerative immune signaling. There's angiogenesis that's promoted in the tissue to provide blood flow and substrate delivery. And ultimately, this allows the tissue to have its normal function in sequestering excess energy and helping to preserve systemic insulin sensitivity. Now, under certain conditions, and this is also heavily influenced by genetic factors, as we've learned over the past decades, conditions like obesity, aging, as well as deficits in adipose growth can lead to what we think about as a pathologic remodeling of the adipose tissue. And so in this state, what is commonly observed is a diminished capacity for new adipocyte generation from precursor cells. So a diminished differentiation process. And that's associated with the growth of individual fat cells. So each individual fat cell gets larger. That's adipocyte hypertrophy. This is associated with and causes cell stress and promotes a pro-inflammatory and pro-fibrotic state in the adipose tissue. And this is highly correlated, and I think the field generally thinks this as being an important trigger of systemic insulin resistance and susceptibility to cardiometabolic complications. Okay, so we have developed a really strong interest now in understanding the role of what I'll refer to as mesenchymal stromal cells, or MSCs. You can also think of these cells as fibroblasts. I kind of use them interchangeably. Thinking about the role of this cell type in the context of adipose tissue remodeling. And so MSCs have played many different and important roles in a lot of different processes in many tissues. They're, of course, really critical as structural cells. They secrete a lot of extracellular matrix. They provide a lot of the tissue structure in many organs, including in fat tissue. And we realize now through the last several years that these cells play an important role in regulating immune cell function, and I'll get into that in the first part of this talk. In fat tissue, of course, they also play this other important role in that they provide really the pre-adipocyte or progenitor for the main parenchymal cell type in the tissue, which are adipocytes. And so now, a few years ago, David Merrick, endocrine fellow, and Alex Sakers, an MD PhD student in the lab, developed a model through single cell sequencing analysis as well as lineage tracing, where they were able to essentially delineate what we think of as a mesenchymal stromal cell hierarchy in the fat tissue. And so they identified a progenitor cell population that's marked by the expression of DPP4, PI16, and these other genes that we think serve as the most sort of upstream cell type in the adipogenic lineage, and that these cells can give rise to more committed pre-adipocyte cells that express PPR gamma, and then these pre-adipocyte cells are highly poised to undergo conversion into mature adipocytes. These DPP4 cells are also interesting in that they have what you could consider to be mesenchymal stem cell activity, at least ex vivo. So they have the capacity, at least, to give rise to a lot of other mesodermal cell types, including bone and cartilage. They're also highly proliferative. So we think of this as a more progenitor-like cell that exists in adipose tissue. The other interesting feature related to these cells is their location in the adipose tissue. So they occupy a distinct niche, and they're mainly located in the septa and fascia that really delineate the lobes of adipocytes. So you can see that here in the electron microscopy. You see this fascial tissue called septae that really surround lobes of adipocytes, these large cells here. And you can see that here. This is staining of visceral fat tissue in mice, and you see the DPP4 positive progenitor cells marking that fascial structure. So a couple of years ago now, Karima Jarina, she's a postdoc in the lab, along with Ryan Calhoun, technician in the lab. We're interested in understanding or asking the question, how does obesity regulate the function and fate of these mesenchymal stromal cells? And then reciprocally, how might these cell types that develop in obesity influence the disease process? And so the model they use will be probably familiar to many of you here. It's the high-fat diet-induced obesity in a C57 Black 6 mouse background. And this serves as I think, a nice model of the pathologic remodeling that happens in adipose tissue. When these animals gain weight, the visceral adipose tissue especially undergoes this pathologic remodeling process. You see fibrosis, inflammation in the tissue, and you see diminished adipocyte differentiation, especially at late stages. And this, of course, is associated with insulin resistance and fatty liver disease, all of the hallmarks of the pre-diabetes state. And so their project started out with a single cell sequencing analysis. This is of the stromal vascular cells in the adipose tissue. They looked at, in this case, the epididymal white adipose tissue, which is a model of visceral fat in mice. And on the left-hand side is the profile of cells under controlled diet conditions, and on the right is the high-fat diet conditions. And this is the profile of all the stromal cells, so no adipocytes here. And what you can see and what has been observed by many labs in the field is the induction of these more pro-inflammatory type cells, including CD9 positive macrophages or lipid-associated macrophages that develop in the adipose tissue. However, we wanted to zoom in and really focus on the mesenchymal stromal cells, and I should point out that's this sort of large pink cluster here. And if you recluster only on that compartment in the lean and obese state, I should point out this is, I think it's 12 weeks of high-fat diet. What you see is that there's essentially a shift in the gene expression profiles and really the profile of these mesenchymal stromal cells in the context of obesity. And so I'll just point out the progenitor cells, that's these DPP4 positive cells here on the right. They diminish in the context of obesity, and we've seen this consistently in many studies. And the pre-adipocyte cells are here on the left. They also decrease in the context of obesity. What we see emerge is this cluster of these blue cells here that we've ended up referring to as calf-like cells for cancer-associated fibroblast-like cells. And so these cells, and the reason we call them calf-like cells is that these cells express all the canonical sort of cluster-defining genes that have been also described in these cancer-associated fibroblast cells, including, as I'll point out and talk about, SOX4 and MDK or MIDKIND as being markers of these cells. And so if you do a pathway analysis of these different mesenchymal stromal cells, you see pathways in the progenitors that make sense, things related to progenitor function like TGF-beta signaling, Wnt signaling, et cetera. The pre-adipocyte cells are enriched for pathways involved in adipogenesis, AKT signaling, and fatty acid metabolism. And the calf-like cells are enriched for pathways related to epithelial mesenchymal transition, inflammatory response that I'll talk about, and angiogenesis. And just to point out, cancer-associated fibroblasts, these are cells that are present in the tumor microenvironment, and they undergo sort of phenotypic modulation in that environment. They have, so they develop from normal fibroblasts in the tissue, and they undergo various alterations, including things related to their metabolism. They express different secreted factors, and ultimately they play a variety of roles in regulating tumor development and growth. And so we wanted to sort of dig in a little bit and understand the induction and role of these cells. And as I pointed out, SOX4, which is a transcription factor, was the most highly enriched marker gene in this population. And so first we just wanted to look at the expression of SOX4 in these cells and look at the induction of these cells over a time course of high-fat feeding. And so what you're looking at here is from 4 to 16 weeks. of high-fat diet, and you're looking at SOX4 gene expression levels within the stromal vascular cells. And so what you can see is there's an induction of SOX4 expression in both the epididymal fat and another visceral fat depot here, the retroperitoneal fat. And then if you do staining for SOX4, you can also see in the control diet, don't see much SOX4 expression in green. In the context of high-fat diet, this is at 16 weeks, you see this really strong induction of these SOX4 expressing cells in the tissue. So there's a progressive induction of these SOX4 positive mesenchymal stromal cells in response to high-fat diet, especially in the visceral fat. Now, we thought that SOX4 as a transcription factor might give us a functional handle into trying to understand the biology of these cells. And so we first wanted to test whether SOX4 plays a functional role in regulating the activity of these cells. And one of the first experiments we did here was to take a control virus, these are lentivirus or SOX4 expressing lentivirus, and induce expression of SOX4 in free adipocyte cells. And so when you do that, what you can see is that if you express SOX4 in these cells, you can induce a lot of the gene program of the calf-like cells. So SOX4 it alone is sufficient to induce a lot of these markers. And this is, I should point out, this is in the fibroblast state, so no induction of adipocyte differentiation. Perhaps not so surprisingly, given that SOX4 altered the fate of these cells without differentiation. If you then induce differentiation of those cells, you see that the SOX4 expressing cells fail to undergo differentiation or exhibit a severe reduction in differentiation efficiency. This is shown here with BODIPI staining of lipid accumulation and a reduction in the expression of fat cell specific genes. So SOX4 is turning on the calf program in the fibroblast state, and then these cells are unable to go efficient differentiation. We also wanted to test the activity of the native cells from the adipose tissue. So we were able to obtain a SOX4 GFP reporter strain. This allows us then to sort out SOX4 positive cells from the adipose tissue. And so we sorted out SOX4 negative and SOX4 positive mesenchymal stromal cells. All of these cells are marked by this common marker, PDGF receptor alpha. And then if you take these cells out of the animal, you can see that this, I should point out, this is from 12-week high-fat diet-fed animals. You see that these cells, these SOX4 expressing cells, have higher expression of these calf-like markers, and they have a defect in their ability to undergo adipocyte differentiation and express markers like adiponectin. And so to then go in vivo to try to test what the induction of this profile or the induction of these cells, what effect would that have on the adipose tissue and on the animal? We've used a couple of different approaches. Here is a gain of function approach where we have a SOX4 overexpression system. So this is a ROSA 26 lock-stop-lock system where SOX4 is inserted downstream of this the stop cassette in the ROSA 26 locus, and then we cross that with a PDGF-free ERT mouse line. So this is a tamoxifen-inducible Cre recombinase that's going to be expressed specifically in that mesenchymal stromal cell compartment. And then upon tamoxifen, the idea is that you get induction of SOX4 specifically in the fibroblasts or mesenchymal stromal cells. The system works really well. We get nice induction of SOX4 in the Cre expressing mice in both the in all the fat depots that we look at. This is the stromal cells. And important to point out that the levels of SOX4 that we see, this is at baseline, so no high-fat diet, are comparable or not out of range with what we see as the induction of SOX4 in the context of high-fat diet. So about the levels that we'd see at 12 to 16 weeks of high-fat diet. And so these animals, I'll just show you the paradigm here. So we induce SOX4 expression in these cells at time 0 here, and then we maintain the animals on a chow diet or regular diet for 4 weeks, and we switch them to high-fat diet for the last 2 weeks. And so what you can see is that there's no effect on body weight. Here, the mice maintain normal body weight and gain the same amount of weight on that brief high-fat diet. And there's no change in adipose tissue weights or liver weights at the 6-week time point. Now, if we look at oral glucose tolerance testing as a measure of metabolic dysfunction and insulin resistance, what you can see is that at the 2-week time point, so this is after 2 weeks of induction of SOX4, there's no difference in glucose tolerance. At 4 weeks, you start to see a sort of maybe mild but not significant dysfunction or worsening of glucose tolerance. But if you give a short high-fat diet treatment, this is 2 weeks, you can see that there's a significant worsening of the glucose tolerance in the overexpression mice. So this suggests to us that the induction of SOX4 alone in these cells, and presumably the induction of these calf-like cells, is not sufficient to cause metabolic dysfunction. But if you have a short-term high-fat diet, so an additional stimulus, there's a synergy between these effects, and that leads to a worsening of the metabolism. And now if you look at the tissue at in these animals, what you can see is that the SOX4 overexpression leads to the induction of these calf-like signature genes in the animals. And what's very notable, and is really a theme for this pathway, is that you get a very strong induction of immune cells, including the induction of macrophages, here shown with F480 expression, and especially T cells, so CD8 expression and CD8 positive T cells. And if you look at the histology of the adipose tissue, you can see the overexpression animals have features of that pathologic remodeling. So you have areas of fibrosis and inflammation in the tissue. It overall just looks like an unhealthy adipose depot in response to SOX4 overexpression in these cells. Now, we also did the reciprocal experiment. So we asked the question, what happens if we now delete SOX4 in these mesenchymal stromal cells? And so we took a LOX-P, a floxed allele of SOX4, crossed it to that same PDGF receptor Cre driver. And in this case, what we did was take, actually, this I think is the wrong. We deleted SOX4 in the context of high-fat diet. And waited for 8 weeks. And so what you can see is that there's no effect on body weight, again, both with gain of function as well as with loss of function. However, the deletion of SOX4 in these cells was able to ameliorate the glucose tolerance and decrease the immune cell profile of the tissue. So you see a reduction in the total number of immune cells, so the CD45 positive cells, which was really driven by a reduction in macrophages as well as T cells. So despite there being no change in body weight, so there wasn't a reduction in fat mass, there's an improvement in the immune cell profile as well as the metabolic homeostasis in these knockout animals. And then the question, of course, an important question for us: what are there things that these cells are doing that might influence this response that we're seeing? And so we performed one of these cell chat or receptor ligand network analysis assays or approaches to look at the secreted factors and receptor systems that might be regulated from these cells. And so if you just look at the right-hand side, this is looking at all of the signals or predicted signals being expressed from these calf-like cells in the context of control diet as well as high-fat diet. Control diet or low-fat diet in green, high-fat diet in the pink bars. And the factor that really stood out to us was this growth factor MDK or midkine, which is a marker gene of these cells and which is very highly induced in the context of obesity or high-fat diet. Now, I had never actually heard of midkine. It's actually quite well studied in the context of cancer and tumor biology. It's a small heparin binding growth factor. Interesting to us, it has several potentially or putatively identified receptors. What's been shown in the literature is that it can act through a variety of signaling systems as shown here. So including Notch2, Syndecan, LRP, and PTP, which is, I think, important in the immune cell aspects. But what's been shown in the literature is that this factor has important roles in regulating neurite outgrowth, various roles in embryonic development, as well as interesting roles in promoting regeneration. It's also been shown to be upregulated in a variety of pathologic conditions, especially as being studied in cancer as well as traumatic injuries. And in general, the consensus from the literature is that this factor promotes tumor growth. And so if you just now go back and look at mid-kind expression in the single cell data, you can see that it's pretty highly selectively expressed in this calf-like population. This is within the mesenchymal stromal cells. If you look at the entire data set, you can see that it's predominantly and almost exclusively expressed within the mesenchymal stromal cells compared to the other cell populations. So highly specific to the fibroblast cells. There have been a few studies looking at the relationship between mid-kind and obesity, and there's at least in this study, a positive correlation between circulating levels of mid-kind. So this is levels in the blood, positive correlation with increasing BMI. So higher BMI associated with higher serum mid-kind levels. We also looked in adipose biopsy samples that we're able to obtain from the Penn Biobank. And so what you're looking at here, these are samples that were obtained from all the patients were obese, their BMI was matched, and we separated them out by diabetes status. And what you can see is that we see higher expression of both SOX4 and mid-kind, these markers of these calf-like cells in the samples from the diabetic subjects, suggesting that there might be some association there. And now, so we wanted to ask whether mid-kind might have a role in this process and in terms of promoting metabolic dysfunction. And conveniently, there's a mid-kind inhibitor that was available. And so, and I should point out that this mid-kind inhibitor works by inhibiting the expression of mid-kind. And so in this experiment, we took mice that were already obese, so they'd been on a high-fat diet for 12 weeks. We treated them with a control vehicle or this mid-kind inhibitor for 12 days and then performed the analysis that I'll show you. The inhibitor worked well in reducing mid-kind expression, both in the tissue, this is RNA levels in the tissue, as well as circulating levels of mid-kind in the blood. Again, similar to what we observed with knocking out SOX4, when we inhibit mid-kind, we don't see any changes in body weight of the animals. However, if you look at just the histology of the adipose tissue, this relatively short-term treatment led to a pretty striking resolution of some of the pathologic features of the adipose tissue. So we see reduction in fibrosis. And now if you look at immune cell markers, including F480, a marker of macrophages, and CD8 as a marker of T cells, you see a reduction in this immune cell profile. So blocking this pathway, both by knocking out SOX4 or inhibiting this mid-kind growth factor, leads to a pretty dramatic improvement in the adipose tissue pathology. And consistent with that, we see a marked improvement in glucose tolerance and metabolic homeostasis in these mice. So here's the glucose tolerance. You see that with mid-kind inhibition, you see an improvement in the metabolic status. And so for this first part of the talk, I'll just summarize here with the model of what we think might be happening. As I told you, in the context of healthy adipose tissue, where you have preserved fat tissue function, you have this pro-regenerative immune cell profile, and you've got adipocyte differentiation, or at least a capacity for adipocyte differentiation through this pathway from progenitor cells to pre-adipocytes and eventually into new adipocytes. in the context of this pathologic remodeling in obesity, what we think is happening is that these progenitor cells, these DPP4 positive cells, are diverted away from the adipogenic lineage. And so instead of making preadipocytes and adipocytes, some of these cells are being induced to undergo this phenotypic modulation into these calf-like cells that express SOX4. We think that SOX4 is a key driver of this process. And then these calf-like cells, in addition to having this diversion away from the adipogenic lineage, which presumably decreases the formation of new adipocytes, these cells also secrete a lot of different factors, including this growth factor midkine, which we think actually acts directly on the T cells to promote this more pro-inflammatory milieu in the tissue and promote this unhealthy remodeling. Okay, so that's the first part of the talk. Just make sure I had time. And so the second part is going to be kind of completely different. So there's no easy way to connect these, I have to say. But for the second part, I'll just sort of explain to you how we got to where we got to. So as Claudia pointed out, our lab has had a long-standing interest in understanding differences and pathways that regulate the development of white fat versus brown fat tissue. And I told you that brown adipocytes are important and are specialized for energy expenditure. And one of the pathways, or one of the proteins, as Claudia also mentioned, we've had a long-standing interest in, is this transcriptional factor called PRDM16 that really drives this process and can convert white adipocytes into brown adipocyte-like cells. And so in a project a few years ago now, part of the project was studying the perivascular adipose tissue. And as part of this project, we had performed a variety of single cell and single nucleus sequencing analysis. And this is an example of looking at human periaortic brown fat tissue. And so what you can see here is this is single nucleus, so we captured the adipocytes in this case. And if you look at PRDM16 expression, you can see PRDM16 expression here in the adipocytes, and there's actually enriched expression of PRDM16 in these UCP1 positive corner of this profile. I should point out that we call this a brown fat tissue because it has a potential to be express UCP1, but really you only see those thermogenic features like UCP1 in the context of cold exposure. And this is from humans that weren't cold exposed. So you see a few UCP1 expressing adipocytes, and they're enriched for PRDM16 expression. Now, the other place we saw high levels of PRDM16 expression is this cluster here, this small cluster, which corresponds to smooth muscle cells. So these are smooth muscle cells that are marked by the expression of MyH11 and other canonical smooth muscle cell genes. And so we got really interested in what the function of PRDM16 might be in this cell type. And I also should point out that a lot of GWAS studies now have identified variants in PRDM16 that are associated with coronary artery disease. And you can see those variants here on the right. And in general, there has been sort of, at least on the genetic level, a strong signature with cardiovascular disease and less of a signature with metabolic traits, including BMI and diabetes related traits. And so just to go back to the expression of PRDM16 now in smooth muscle cells, and I'll point out that Josephine Tan, a postdoc in the lab, has really conducted all the studies for this second part. And so if you look at PRDM16 expression now in human tissue, you can see very highly expressed in arteries and from different areas. And you can see some expression here in the stomach. If you look at single cell analysis of the vascular compartment, you can see that the expression of PRDM16 is highly enriched in the smooth muscle cells as compared to the other vascular cell types, like endothelial cells and fibroblasts. And in mice, you essentially see the same thing. So here you're looking at expression in the aorta, you can see that there's massive expression of PRDM16 in the aorta compared to where we've been studying it in the brown adipose tissue. And again, single cell analysis would say that the expression of PRDM16 in the vascular bed is mostly confined to the smooth muscle cells. Now, smooth muscle cells turn out, I was relatively ignorant at the outset of this project, they turn out to be a really interesting cell type, especially from a developmental biology perspective, in the sense that they can acquire and undergo a lot of different sulfate transitions. And it's been shown in the literature that they have the ability to make adipocyte cells, macrophage-like cells, and the various cells shown here. Now, this is particularly of interest in the context of disease conditions. So for example, in atherosclerosis, smooth muscle cells undergo this classic phenotypic modulation or switching, or also referred to as dedifferentiation, and really give rise to a few different cell types, including fibroblast-like cells or synthetic cells, as well as macrophage-like cells and osteogenic cells. And this is important because at the end of the day, in a developed plaque, in a mature plaque, about 40 to 70% of the cells within the plaque are derived from smooth muscle cells. And that's been shown from a number of really nice lineage tracing experiments. So just as a sort of general introduction, in the context of atherogenesis, of course, the initial stages, you get injury to the endothelial cells, you get accumulation of lipid, inflammatory responses that sort of trigger the onset of the plaque development. The smooth muscle cells, the canonical sort of contractile cells, live in this media layer of the vessel, and they are induced to become activated, they proliferate, and they migrate into the intimal space and differentiate into these different cell types. And one of the key cell types that they become is these synthetically modulated cells, as I'll refer to. And that's important because they generate this fibrous cap that surrounds the necrotic core of the plaque and really protects it against rupture, which of course is a huge problem. And so the point I want to make is that the features and the differentiation of these smooth muscle cells into different cell types is of huge clinical significance because of their important role in regulating the plaque structure. Now, if you look at PRDM16 expression in the context of atherosclerosis, and this is single cell data from Murdoch-Riley's lab at Columbia looking at samples from plaques. And I just wanted to focus in on the smooth muscle cells. They identified 3 different subtypes of canonical or quiescent or contractile smooth muscle cells, these 3 clusters here, as well as this modulated cluster that's disease specific. Now, if you look at PRDM16 expression, it follows essentially the expression pattern of contractile genes, so highly expressed in the quiescent state and downregulated in the context of these modulated or synthetic cells. So it's downregulated during this dedifferentiation process. And so we were interested in understanding the role of PRDM16 in the smooth muscle cells and really in this atherogenic process. And so we developed smooth muscle cell specific knockout animals. And so this worked well. We could delete PRDM16, as you can see here, by staining specifically in the smooth muscle cells as compared to the endothelial cells. This is in the aorta of mice. This manipulation did not change body weight. It didn't change any metabolic parameters in these animals. Everything that we could see from that standpoint was normal in these mice. However, when we looked at the arteries, and in this case, this is the aorta, and this is just bulk RNA sequencing of control and the knockout animals. This is under basal conditions, so no disease conditions. These are just adult aortas from adult mice. What we see under these basal conditions is in the knockouts, there's the induction of these synthetic processes. So all these processes that are associated with that modulated state, that synthetic conversion, are upregulated in the aorta of these knockout mice, again, under no disease conditions. If you look at a single cell level, there was 2 possibilities, right? You could see a small number of these synthetic cells might be arising that would explain that bulk RNA sequencing result, or we may see the induction of the genes across many of the smooth muscle cells. And what we see from single cell analysis is the latter. So if you look at the smooth muscle cells, there's again 3 different subtypes. This again, no disease conditions. You can see that the synthetic genes denoted here with ankyrin RD1 as an example are highly induced in all of the smooth muscle cells. So we're not getting a specific subset or a small cluster of synthetic cells arising. It's really repressed. PRDM16 is repressing these genes across all of the smooth muscle cells. And you can see that here, just looking at all these clusters and various genes, that these are induced across all of these different populations. Now, then the question is, what happens during in the setting of disease? So what happens in the PRDM16 knockout animals? And so the model that we used was to induce atherosclerosis through treating the animals with an AAV expressing a gain of function version of PCSK9, and then subjecting the animals to 12 weeks of Western diet, and then phenotyping the lesions. This system worked really well. We didn't see any change in body weight, but cholesterol levels went through the roof with this treatment, and we got atherosclerosis development. And so we quantified first the lesion area in these animals. We did this histologically. This is looking at the arch region. And so basically what we found is that we didn't see any change in total lesion area in the knockout as compared to the control mice quantified this way. However, what we did observe is a really dramatic change in the structure of the lesions, the plaques that develop. So in the control animals, this is after 12 weeks, you see this sort of canonical appearance of these plaques. They have this sort of foamy appearance. You can see a lot of lipid within the plaques, whereas the PRDM16 knockout lesions look really unusual. They're highly fibrous, a lot of smooth muscle cell content in these lesions. And if you stain for collagen, you can see a dramatic induction of collagen, as shown here with trichrome staining or serious red staining in the tissue, and that's quantified on the right. So the knockout of PRDM16 is leading to this highly fibrous and collagen-rich lesions. Reciprocally, we see a reduction in the lipid content. So this is shown here with staining for PLN2. Controls have a lot of PLN2 stained here in green. And in the knockouts, there's a really dramatic reduction in PLN2 positive cells. Now, what I showed you in that last 2 slides was a constitutive deletion, and smooth muscle cell selective, but present throughout the life of the animal. If we do a more acute knockout just before inducing atherosclerosis in these animals, we get similar features, although a little bit less dramatic. And what we see in this case is with inducible knockout of PRDM16, you get lesions that have very prominent cap-like structures, so still more synthetic cells, more collagen content, and similar features, but they don't have that super fibrous appearance that the constitutive knockouts have. And then we wanted to do the reverse sort of experiments and go try a gain of function approach. And so to do this, we took human smooth muscle cells in culture. And I should point out that PRDM16 expression is decreased as soon as you take smooth muscle cells from human or mouse out of the animal. So the expression of PRDM16 decreases immediately. And so it provides a nice gain of function system to ask about the activity of PRDM16. So what you can see here is that overexpression of PRDM16 in these cells reduces the expression of many of these synthetic genes. And functionally, you can see that overexpression of PRDM16 suppresses these synthetic processes, including reducing migration in these cells as well as proliferation. So it's sort of dominantly repressing a lot of the synthetic activity in these smooth muscle cells. In terms of mechanism, we're still trying to work out the details. what I can say at this point is that we know that PRDM16, at least in the cell culture models, is binding directly to many of these synthetic gene loci, and it's modulating the chromatin state at these genes. So it's leading to less active sort of marks of chromatin at these genes. So we think it's acting directly in chromatin to suppress this gene program. And so just to end on the second part, the model where we currently stand is we've shown that contractile smooth muscle cells or so-called quiescent smooth muscle cells have high PRDM16 expression. PRDM16 expression declines during this modulation process in the context of disease, including atherosclerosis. And that's associated with the induction of this modulated or more synthetic phenotype. And then mechanistically, we think that PRDM16 is binding to these genes, many of these genes. We're trying to understand all the components of the mechanism. We think that it's being recruited by various transcription factors. RUNX1 might be one of these transcription factors that's recruiting PRDM16 to these genes. And I think the idea that we have is that PRDM16 is forming a repressive complex at these genes, and it's essentially shielding the genes from the activity of these pro-synthetic or de-differentiation cues, including PDGF, TGF-beta, and presumably others, and sort of blocking the synthetic program, preventing that from being expressed. As soon as PRDM16 expression goes down, those genes can now be expressed, and that modulation process is able to be realized. And so I'll just end with probably the most important slide, thanking the people in the lab. I pointed out the people that sort of drove the projects, Karima and Brian for the first part of the talk, and then Josephine somewhere in this picture at the back for the PRDM16 athero studies. We had really amazing collaborators. I'll point out Veronique Lefebvre, who provided a lot of our SOX4 reagents for the studies, Murdoch Riley at Columbia, and a really nice collaboration with Esther Lutkins for help with the atherosclerosis studies. And thanks everyone for listening, and happy to take any questions.
Questions for Patrick? Let's start with the first one. So when you look at the transcriptional program regulated by PRDM16 in smooth muscle cells versus, let's say, a brown free adipocyte, for example, how does that compare? Are they still both regulating metabolism, or is it just completely different?
Yeah, that's a great question. Actually, no, it's very different. So initially we were thinking, or had the mindset at least, that PRDM16 might be regulating smooth muscle activity via regulating metabolism, including fatty acid oxidation. We see no evidence of any changes in fatty acid oxidation, which is one of its key roles in brown fat. And the target gene profile just very different. So
do you think it's going to be a different transcription factor in this case?
I think it's I should say from the motif analysis, there's no evidence of things like PPR gamma that we see very dominantly in the fat cell system. So yeah, I think there's a different complex forming that's regulating these genes. Yeah. Yeah. We had expected to see some overlap and just don't see that.
Tim?
I'm not that loud, I swear. Really nice talk. So I have a question for you about the mid-kind timing. So I might have missed this, but when do you first start to see that go up? I mean, it seems like at 8 weeks you start seeing SOX4 levels start to come up.
Yeah.
And it's probably corresponding with that's the first time point. Is it earlier? Because I'm curious about this connection with T cells, which might infiltrate later in that model, and whether it might be working on myeloid cells
earlier.
Yeah,
so that's a really interesting question. We've thought a lot about this. I think so I didn't really talk about it here because it's certainly going to be much more complicated than I presented. And I think it's going to be really interesting. So our sense is that, so midkine levels definitely increase and are present before there's a strong, pro-inflammatory profile. And our sense is that it's having some adaptive roles at early stages in adipose tissue, including potentially promoting adipocyte differentiation. And so those effects are there. I think we don't have all the details, but it's probably working through different receptors and certainly different cell types. And so it's having some roles that I think would be adaptive and helpful, but that at later stages, our sense at least is that the immune cell roles and this, especially the T cell activity, is dominant. So that kind of takes over, even though there's probably some beneficial effects it's having on some cell types. The T cell effect is dominating at the end of the day. And that's, I think, accounting for why we see an improvement when we block the pathway. But it's working through different receptors, we think, so it'll be kind of interesting to try to tease those apart and see if we can figure out contributions of some of the other components. So definitely where we're headed with this project.
Concerning the transfer of the smooth muscle cells, endothelium, the saturated and polyunsaturated and saturated fatty acid consumption, that is the cholesterol in the blood. You didn't mention, you mentioned about adipocytes, but the thing is that in the circulation in the blood, what are the factors that reduce the smooth cell transfer through the endothelium regarding the fat lipids in the blood, and what is the best fat lipid to consume, that is polyunsaturated fatty acid, to reduce the risk?
Yeah, we haven't not investigated that. So we've only looked at the, I mean, in our model, we induce the fatty, the levels of fat and cholesterol are induced to the same level. And so the initial events that cause the injury to the vessel are, we assume, are similar in both the control and the knockout situation. And it's really the smooth muscle cell activity that's being modulated. Now, how some of the initial process with how the smooth muscle cells migrate out of the media into the, into the intimal space. We also have not studied that process, but some of that is known from the literature. Yeah. I don't know if I answered your question, but.
Yeah, what?
Pardon me?
It is worthwhile to study. I mean, you haven't done studies, did you?
Yeah, no, it's
definitely. Yeah, if you do it, I mean, it's worthwhile because it's something. When we eat, whatever we eat plays a big role in the health of our vascular system.
Agree. Totally agree.
Great talk. My name is Utkarsh, and I wanted to ask you that is cancer-associated fibroblasts that you saw it in the adipose tissue. Do you postulate that it'll be just be induced via diet, or could it be seen in other instances like aging or lipodystrophy or other metabolic syndromes?
Yeah, that's a good question. I mean, in the context of aging, we also see the induction of the signature and the cells in adipose tissue. Aging is a little confounded in a sense that in addition to aging, the animals are more obese too. So we think that probably the obesity is the driving factor in that case. We do not see the induction of the cells in other organs like muscle, for example. So in the context of obesity, so we think there's something. I mean, we know that adipose tissue is, at least at the early stages, is kind of becomes very stressed in the context of high fat diet. So there's something selective to adipose tissue in terms of like what the signals are that induce these cells. We don't know exactly. We think TGF beta, which is always kind of easy to point to, seems to be involved in almost everything, is one potentially driving factor. But then how that's induced and which cells are producing that or how it's regulated, I think is still unclear. Yeah.
Thank you.
Great talk. I had a quick question about the atherosclerosis. Did you look at males and females by any chance? Sorry if I did missed that. And if you did you also look at the aortic root of these of the lesion as opposed to like the sectioned aortas?
Yeah, that's also, so first of all, we did look at males and females. I didn't show the female data. You were looking only at males. The female data, the extent of atherosclerosis is different, but the effect of Pyridium-16 is the same in males and females. So that's conserved. The aortic root we did look at, it's quite different actually. There's no Pyridium-16 expressed in those cells, so we don't get the same effects in the root as compared to the other parts of the aorta. So it's actually pretty interesting. Yeah.
All right, well, thank you very much, Patrick. Thank you.